Okay, and good morning to all, and thank you very much for joining this webinar. And as you saw in the video some time back, so we will walk you through the whole solution portfolio from the genomics part to the drug discovery part, which will involve systems biology approaches and different molecular modeling and simulation approaches that speeds up or accelerates the drug discovery process. And the whole process can definitely be applied to the other sectors where a molecule design or molecular interaction plays a very key or important role in innovation. So it could be in the food, it could be in agriculture, it could be partially in chemicals, it could be partially in other different sectors of microbiology, biochemistry, molecular biology, pharmacy, and so on. Much beyond what we can think of. Okay, that's my initial slide. And we'll start with the challenges and how do we mitigate these challenges. That is extremely important. So the concept of pharmacogenetics and pharmacogenomics is extremely important because any variability in the nucleotide sequence will definitely affect the drug efficacy, and that's where our personalized medicine plays a very important role. What medicine may be good for me might not be good for the other person, and so on. So how do we make it? The goal is to select the drugs with likelihood that it benefits rather than giving harm to individual patients based on the genetic makeup. The genetic makeup could be based on a territory, based on a geographical index, or it could be based on a community basically. So these are some of the relationship that we need to study, and this relationship, there will be a conserved polymorphism in all of these relationships within community or within a country or within a geo, and how do we investigate that at the genomic level. And that's one of the challenges because of the huge set of data. And these drug targets are categorically divided into three different categories. One is a direct protein to the target, and which we have been working predominantly on. Other one is single signal transduction cascades, or the downstream proteins what we call. Other one is disease pathogenesis protein. So these pathogenesis proteins may come from proteomics, may come from some of the images of the tissue and things like that. A radiologist or a pathologist or hematologist may also give his put of inputs in identifying these proteins or these tissues from where the proteins can be expressed or identified. So, given that a drug responds to a large number of proteins, and all of these genes have certain or specifically several polymorphism associated with them. So, a single polymorphism on a single gene would not express the variability of the drug responses basically. So, it is both totally polygenic, or a genomic approach should be the best way where we address these types of issues. So this investigation, what we are going to do or whatever I'm going to showcase now, has the capability to enhance clinicians to use medications, personalized drug discovery approaches, so that it will give a good clinical potential to the discovery or to the research or innovation that goes by. So these are the challenges, and how are we going to address these challenges based on these points is what we are going to do. To address these challenges, Dassault Systems brand BIOVIA offers a series of solutions. Although all of these, because this is totally interdisciplinary, so we cannot rely on either the biology or the imaging or the chemistry collection, but combines together and works in sync. So chemistry collection plays a very important role on that at the small molecule level. The biological collection is used for identification of biomarkers, quantification of protein biomarkers, NGS data analysis to find variants basically, to identify genes, insertions, and so on. And imaging collection works at the diagnostic level, either at the diagnostic level or at the assay level, where you can have your neurite labeling assay or NFKB translocation assay, where these results are extremely important. Even a simple extraction of the protein is also important from Western blotting techniques basically, where you can bring the protein on the nitrocellulose membrane and then quantify the protein based on the spread or based on the outline of the protein on this nitrocellulose membrane basically. So these are the different approaches that goes by, and we will discuss some of them with some of the examples. Now, in the NGS, in the next generation sequencing, yes, it has advanced a lot, because in 1980-85 when first they started sequencing the human genome, it was 20 plus years that took to sequence the human genome. Today it can be done in an hour, couple of hours time, where you can sequence the whole genome. Sequencing is equally important, but data analysis also is more important, because what you do with this data, that is extremely important. And if you're going to use some of the conventional methods like command line coding to analyze NGS, so you can imagine that you need to have a sync, or you need to have a follow-up of the result that comes from one of them. Say from the quality check, basically fast QC, you will go on to analyze is the next level of, say, making a mapping basically with the reference genome, or using a Dno Assembly. So that will constitute a set of codes. So you can come out with a workflow sort of thing, but that will definitely demand some coding to be done, and a concept needs to be defined. So that is one of the challenges which is there as far as the next generation sequencing data analysis is concerned. And if you're going with some databases or some programs specifically for one application, like for exome sequencing one application, for de novo another application, like that, which are either commercial or academic, then how do you standardize that? Not only the data formats, but also how do you connect to these different sources of programs basically to come out with one approach basically. So that deviation can be mitigated, or that challenge can be overcome by using the BIOVIA's repository system. So this is a very unique approach that BIOVIA's programs have taken up. So that is basically called as the repository system. It's like a box basically, where you have all the data, and whenever you want, you query and pick the data, and all of them is GUI based. But of course, we are not denying that people who wants to do a command line approach to make their own set of algorithms or modification of the existing algorithm can also do it. So this enterprise platform will allow you to seamlessly acquire data, perform modifications, analysis of this data, and deploy the report as per your requirement. So all these is possible with one solution that it goes with. And once the gene expression is identified, you can take the sample and then run through the proteomic studies, or some of the imaging studies basically on the MALDI, before the MALDI, the 2D page basically, and analyze the quantify the protein also. So that is also another set that will follow through this. So all comes with one solution that offers. And following this, when you have identified the target, so I told you about 3 types of target, and if you have identified this target, and if you are looking at identifying creating an antibody as a monoclonal antibody as a solution to your disease pathogenesis, or if you're looking at a small molecule to inhibit the activity of the protein from the pathway, or if you're looking at a protein-protein interaction or an intercalating protein-protein interaction, we can achieve that. So what we offer you is a complete package, and you have the liberty to choose what you want to do. If you're securely working on NGS, yes, you can do only NGS, and other things can be is shared across different departments who they are working on. That's the greatest advantage that you have, and everything will be seamlessly on one platform, showing no deviation, or the standardization will be complete. I'll start with the NGS part, the next generation sequencing. Although there are many instruments which actually provide you the NGS data, the capability of the solution is to access all different types of data, basically the read data, I mean. So we have experience working with many of our customers. One of our customers is working with horse, basically the equines. So they are studying the equine genome basically, and the pathogens associated with the equines basically. In fact, we were working with them on that using this powerful NGS tool, and they are working very strongly on that. We have some organizations who have been using this for the last 15 years, and in Singapore and in England and many other places where they are using this. So the concept is very simple. I have a powerful repository which is totally flexible, and all of these programs, they actually seamlessly communicate to this repository. So one typical thing is that every researcher are highly innovative, and every professor who works on this tries to implement innovative ideas. What we do is basically we are supporting them. So if you are using a program called Breakdance for alignment or something like that, so you can definitely integrate those programs within this application. So what we wanted to basically say is we work along with other programs. We are not competing against the other program, but there is a sequence that is already available, which will facilitate for you to move forward faster with with a defined workflow. That is what we offer in this. I'll show you some of the examples as we go on. Other one is quality assessment and processing. Depending on the averaging of the reads, basically average depth of the reads, you can ascertain what is the quality of the read. So we use this fast QC program here, and fast QC program can be used at two approaches. I'll show you in my video when I run that how we can do that. And we also have a capability to perform mapping. So mapping across in RNAs, in de novos, in gene sequences, genome sequences, basically we have different approaches, and all these programs are tightly integrated with this particular program. So you don't have to download something separately. I don't have to download Bauti or Velvet separately, write the command, integrate. All of them is gone. Everything is present within the program. All you need to do is add the set of parameters that you want to add. Then we have a variant detection, profiling, and comparison. So you can identify SNPs, you can identify insertions, deletions, you can identify copy number variations, and structural variants. So that the program is called Breakdancer, that is also integrated with the program. So any other program outside of this can also be integrated. You can do automated comparison among the samples and the different feature sets, basically. So these are the things that you can do from the variant detection part. And then you also can work with RNA Seek Analysis. So I'll show you some of the examples on that, where the expression patterns and the transcript structures using cufflinks, cuff compare, cuff def. These are the different programs that we have, and these programs are tightly integrated with the solution that we will be offering. And also, of course, the chip seek analysis, where you can detect the enrichment peaks, basically our transcription binding, factor binding in patterns. So it's integrated with cis genome and max respectively. So these are the different programs. Of course, the visualization can be done by gbrowse, can be done by NGS browser, and so many other visualizations are also exist within the program. So you don't have to download anything outside. In fact, even the genomes that you're working on, you don't have to download it separately from the FTP sites from NCBI or any other site. So we have links, everything which are integrated within the program, and you can download it. For example, if I'm going to work with, say, example, a plant protein, and then I don't have the genome on the NCBA, the corresponding, but I want to work with plant EMBL data, then I can actually add a link to this particular program, and that link will automatically prompt me to download or give me an options to download. So I can customize as per your requirement. So this is the different ways by which the NGS plays a very important role, and in analyzing the NGS data and coming out with wonderful results. So we have many customers who are working on this, and they have been using this program predominantly. Then what is the approach that the solution takes in? So it's a data management system, basically. You can do parallel processing, all of this can be done for aggregation, annotation, analysis of NGS data, basically. So you can have genome sequences, you can have GFF files, the feature files, you can have map rates, you can have BLAST databases, and many of the other analysis items. We have integrated with CleanWare and DBSNP, CRISPR, ONT. All of these programs are present within the program. I will show you a snapshot of them in the slides as we go on. So what are the advantages if you're using a solution from Dassault Systems BIOVIA for NGS analysis? What are the advantages? You can eliminate data management headaches, basically, where data may lie in multiple places. You want to pick data from one place, then find out the file paths associated with that, then run this program using your command line. All that headache is totally gone because you have a browse button by which you can pull in the data. But I'm not limiting that to that. It's easy to use because some people would definitely want to implement their ideas and try the difficult path out, because there might be some innovation that comes out of that. So that capability is also present within the program. You have a complete flexibility here, so you can add, remove repositories, you can add repositories, you can rename old repositories, add new repositories, perform mapping. So once you create a repository, that repository will be with you. You can make all sorts of modification within the repository itself, so you don't have to go back and start from stage one. That is the advantage you have. Then you have rich annotation options, basically. So if you want to tag some additional information with respect to a repository, for example, you want to add some tumor reads, basically, or from a sample, from a normal sample, or from a tumor sample, you want to add some reads into the repository, you can add it so that you can query that also at any point of time. So this type of annotation options are also existing within the program that allows you to annotate. You have unlimited linking options. So if you want to link, say, a feature, gene feature, or a CDS sequence with a repository information with related to a mapping experiment, then you can do that by setting it up. So all of these things can be done. So linking options are available. And this is a powerful system because it is based on query. So as we know that when we query databases, it is typically very fast. But when we are going to search, there is a difference between searching and querying, basically. And what we do here is basically querying. So once a repository is populated and you have all the details in that icon, it's just a box of information that you have, and you can pick any information at any point of time. We have also machine learning integrated within this program. So if you're looking at doing some machine learning with this program, you can also do that. So this is an integrated data analysis and management solution that we'll be offering. So you can retrieve queries, you can update queries, you can update data. All of these things can be done. So there is no locking in, so which means that the different file formats are supported in the form of readers. And these you can also write them in all these formats. So if I want to write a file, sequence file in FASTA format, I can do that. So all of this is possible with just one writer. That way it allows you to do it. And it's totally flexible, which means that there are some custom components which are available. So fast QC, there will be one component for fast QC, and then you just browse and add the reads and it will do all the analysis when you run it. I'll show you on the run now in a short while. So this is the ease by which this particular program works. Now you have seen what our approach is and what are the advantages that you have when you use a solution from Dassault Systems BIOVIA. So the next one is basically the different analysis that you can do. Okay, sorry, I'll take up the questions. You can type the questions on the chat. I'll take up in between, basically, because I'm seeing it on the full screen when I'm explaining it. Okay, so the NGS capabilities, what we have. So you can have the analysis part where you can do genome analysis, you can do mapping using different programs, you can do RNA seek analysis, variant detections. We also support the best practices from GAT toolkit also, GATK toolkit also. ChIP-seq analysis can also be done with the program. Methylation uses Bismarck tool, which is totally integrated with the program. Dno Assembly is also part of the program, which uses contigs as reference sequences when you don't have a reference genome, basically. RNA seek is associated with Top Hat, Cufflinks, CuffCompare, CuffDef. All of these programs are embedded within the program, and the sample programs are all available. So To start with, you can start with what we offer actually inside the program. So we have around 70 to 80 different protocols across all of these. So you can just feed your data and then run it and get the required results. In fact, we have worked with many customers on this. We have a CRISPR editing tool also, where you can edit these nucleotide sequences using the CRISPR approach and then go for mapping and other sequencing analysis. So for the variant detection, you have CNV variant detections that is possible, and you can compare different experiments. So what happens is basically when you create a repository, add the features, add the reads inside that GFF file, then the reads FASTQ file. It takes even in the zip format, so you don't have to unzip it also. So when you take it, and then what happens is when you are querying it, it creates an experiment that you will be naming it according to your words basically, and that experiment will be reflecting in all the study that you can do. That's why I said you can have multiple experiments within one repository, you can have one experiment within the repository, so or multiple repository, multiple experiments. So these approaches can be taken into consideration. Then you have the GATK best practices. So the best practices to call variants is available within the program. There are 5 different protocols that's available within the program that allows you to do it. Resequencing is another important aspect where you want to see the distribution of mapped reads across different chromosomes. So if you want to do that, you can do that with this program. You have viral population analysis to find positions that have variations in a population. So when you're working with a sample and a population, you can do all viral population analysis also. And not only that, many of our customers typically started with using the standard package, which involves all of these capabilities, and every year or every 6 months we actually upgrade new programs into this. And in fact, now we in the next release you will be getting something related to more related to the genetic pathway and other things coming up in the program, so which we are actually working on, the R&D is working on, and you will get it soon on that. So these are the different capabilities as far as the NGS is concerned. I'm going to show you one example. Basically, you see I'm using an E. coli here, and I'm just creating a repository for E. coli. What I'm going to do is, you can see that I have a FASTA sequence reader, and I create a sequence repository. There are multiple options where I can create. I can and add the name of the repository. Then I have a GFF file, basically the feature file. It supports GFF2, GFF3, and the general GFF. And then I name the, all you have to do is when you are actually creating the repository, you make sure that you're giving the same name. So this is the repository that I have created from my standard file, and that has the gene information, that has the CDS information, and so on. Now I have the reads from Ion Torrent or Illumina or ABI Solid, whatever be the instrument. Okay. So I have the reads, I'm picking this reads, and I have just one component where I can browse and pull in the reads over here. What it basically does is, sorry about this. Okay, I don't know why this came up. Okay. So you get the fast QC report. This is the conventional fast QC report that you get when you actually run on the web using this particular program. And this has the whole information related to the a report of the reads basically, and you analyze this reads and then come out with a conclusion saying whether the quality of the reads is good. Can I go for an analysis, or do I need to actually provide a new set of reads for me to work out with the new set of data? So these are the information that you can get it directly from this. The other option is a program which is inbuilt within the solution itself, and that is basically a QC analysis which is there. I will show you that shortly. But before going to that, if you want to do this mapping, basically I have analyzed the reads, I'm going to do the mapping here. And what I'm doing is basically I had already the repository, and I had the reads also associated with that, which I did the quality study on that. And these reads could be paired reads or unpaired reads, so both options are available for me to upload a full directory of reads. So sometimes I may have one read, a big file, I may have multiple reads. So all of this information can be uploaded from the directory. So it's totally optimized, and it will not be as slow as we do it conventionally, but it will be quicker because you can do parallel distribution of these analysis also. Then you have the mapping program which is there, which uses BWA, and in this, so you can see that there is something called an experiment name. Basically, that is something you will define it. So going forward, after the mapping, when you are actually querying it, identifying the SNPs or genes or features, that is what will come out from the that will be constantly asking you what is the experiment that you want to work. That's why I said you can have multiple experiment within a repository, you can have one experiment within the repository, and so on. Multiple things can be done. So once you do this, what you basically get is the total details related to the quality mapping as well as the coverage and the reads that are mapped. So you can see that the coverage of mapping here is 98%, which is extremely good. The depth information is given, and the tabulated information related to the repository also is provided inside that. So first was a QC from the fast QC. The second was a QC along with the mapping that we have actually done. The next step is basically we are going to detect and add variance respectively. So always it starts with all these programs are inbuilt within the solution. All you have to do is just change the reads from E. coli to your reads basically and define this thing. Now you can see that I named an experiment. That experiment is automatically updated. So if I have 10 experiments, I can put the 10 experiments. And what I'm going to do in this is I'm going to identify the SNPs and indels basically. And I've done that, and then some filtering procedures basically, what should be the SNP gap, what should be the indel gap, because that's how it works, right? I can't have indels every base or every 2 base or every 3 base respectively, so there should be a pattern associated that we're going to do that. And once we do this, what happens is the result is automatically goes and sits inside the repository. So next time whenever you want to query this with the new results of the variance, that also will be used in this. So I want a variant between this region to this region which has an SNP of this, which associates with the gene. Very simple, you can actually just query this and get the results of that. So this is how you get a tabulated output of this. This is a simplistic report that we have generated. It's all at your behest that you can generate a tabulated result, or you can generate a graphical result, or you can notify a result based on the threshold that you will be providing. So once all of this is done, I need to do the visualization part. So the visualization part can be done using the tablet, and this tablet allows you to visualize, see the SNPs across the mapping experiment from with respect to the standard genome or the reference genome, as well as the different reads to which it is actually mapped. So this way you can find the SNPs. What you saw earlier was in the tabulated format, and what you see now is in the graphical format basically. And as you know that each of the color represents the level of what is the type of nucleotide associated with that. So green represents adenosine and so on. Thymine is represented by blue and so on. So this is how we can scan through each of them using this. We also support NGS browse and GBrowse too. So if you want to visualize that in that particular tool, you can also do that. So this is how the visualization is done with the SNP identification. Once all of this done, the next step is to identify the SNPs that are within the genes. Basically, that is the important part, right? The exonic part that you want to identify the genes, and then to find the SNP associated with the gene. Then only you will find what is the translated part which is there in the protein basically. So this is just run for 2 of them. So in this we have a couple of genes over here, and these genes basically from the repository, and you see that there is an SNP at the position 1098949 and 1134629. So you see that there is one nucleotide which is changed over there, which may reflect in the expression in the protein at the mass spec level or at the sequence level, which we will analyze going forward. So this way we can get the SNPs within the genes using this particular program. The next program that I'm going to show you on this is the Dno Assembly part of it, where I'm going to use the same repository, E. coli. In this, basically what we do is we create contigs. We are not going to actually support with reference genome over here. And then once we do this, you will see that you can define the regions and the mapping can be done by Mira3 or Velvet. These are the two programs. And as I mentioned some time back, we are unlocked program, so which means that there is no locking in, and you can save these files in different formats. So you have seen that you can save it in a FASTQ writer. So we have assembler for Mira3 and assembler for Velvet, which is present within the program, and that generates an output, which is the assembly reports that you get. So you can see that on the Dno, you get the total report over here, which I'm not analyzing it now over here, but it gives you the full-fledged report that you have. Once that is done, you have to also check the assembly quality by using what is called the mapping approach. So you use your reads basically. Now whatever reads that you have where you want to run this your de novo, just browse and pull in the reads over there. And once you pull that reads in and pass it through this program and run this particular program, it could be unpaired reads, it could be paired reads, and of course then you have the mapping, and then you have the repository query basically that you do. And as I mentioned some time back, whatever analysis that you do or whatever you pull in the reads or the genome or the feature file, all of them will go and get itself, it will sit inside the repository. You have the ability to append the new reads, you have the ability to replace the existing reads. All of this is possible when you choose the option. It's all graphical, and you can do that. So you can see that this is the mapping depth comparison basically with the assembly and after the mapping respectively. And you also have a capability to perform the other one, which is the mapping comparison basically. So all of this can be done within this particular program, which allows you to use graphical approaches to study this. So any depth comparison, depth difference, both of them can be done within this program. So going forward, if you see in this, so this is the difference of the map depth basically. So that information is there, and which shows here, it of course a minimal amount of depth, the difference basically across each of the sequence position is what you see on this program. So this is about Dno Assembly. You can build programs on top of this Dno Assembly, and that will definitely help you to innovate and accelerate your research faster. The next one is basically on the RNA Seek Analysis. So there is a comprehensive help associated with each of these programs, and each of these programs you can take the help which is available. So will give you all the information related to what needs to be added in each of these parameters, and what is the program that it does, what it uses, and what has to be run before that. So what we are going to do here is we are going to create a mouse repository, and we are going to add the RNA seek reads. So typically you can get these reads from your NCBI or any other source. You can download it directly from there, or you can just put the read number, SR number, and then read that too. So this way you can add the RNA sequence reads to this mouse repository. That's what we are doing over here. And you have the whole set of parameters that you can automatically populate, or you can save them so that it can be used as a template from time to time. So you can see that these are the different ways by which you can do. Because it will take some time to run it, and I want to make the video shorter, that's why I was cutting that and showing you the output results basically. Once that is done, I can add the transcript features to these repositories basically. So I was mentioning about cufflinks, the transcript assembler basically, and that is a program that it uses here. So you have to also, I want to also mention that you are not going to install a program separately. All of them, when you have the standard installation of the solution, all of them will be taken care, and you will see the start position, the type of feature, the end position, and all the information related to the transcripts are also available. And if you want to see the mapping depths associated with the transcript, that also can be visualized using this particular program. So all of this is possible within this particular program to run. And if you want to compare the abundance of a transcript basically within the repository, so the same mouse repository I'm going to take, and I have 3 experiments here. And if I'm going to unify all of them basically I'm going to unify all the transcript that's called as cuff merge. Cuff difference will be finding difference like that. All of these programs are available. All I have to do is find union transcripts. Instead of that, I put find different transcripts and then I get it. So you can see this after union, what are the unique transcripts that you have, and you get these transcripts as an output. Like this, you can perform your analysis on the RNA seek analysis also. Going forward, there are ChIP-seq and other things which I have not actually put up as a run over here, but I want to give a very clear indication that yes, we have the whole level of NGS analysis that can be done with the solution. So whatever is your innovative ideas, you can start implementing it, because it's a no lock-in or it's an open and transparent platform that we offer. And when you want to do in the SAC is called sequence analysis collection. Sorry for using that term. We use sequence analysis collection and for all of the sequence related approaches. And each of them is called by a collection. Next will be the chemistry collection. I'll show you shortly. So new CRISPR component exists. So basically what does CRISPR component do is, what it does is basically it is an algorithm to match subsequences which are added as sequence features to this particular data record, and it's based on a publication that we have actually done this. So like this, the CRISPR sequence information is available and the cut regions are available, and that you get it tabulated format. That's the component is available on this. As I mentioned some time back, we have integrated with CleanVar and DB-SNP. So CleanVar is archive of reports of human relationship among variations and phenotypes. So you can download the variant file, VCF file basically, and accommodate it within the program within this particular component and compare that against the output that you get. And of course, DB-SNP is also available as a VCF file where you can pick up your SNPs and then identify whether you have a novel SNP in place or SNP has a phenotypic character associated with that. So all of this can be done using this collection, which is the NGS collection. With the incoming of the Oxford's ONT sequencing platform, basically, so we also have included new parameters, which is the mapping parameter, read mapper basically, with the ONT. So we can read the ONT data. And of course, the ONT 2D reads, if you're going to set it to true, the ONT reads can be mapped with this BWA program. Like that, you can also use count-based substitution, okay, when you want to analyze ONT reads to understand what is a mapping quality related to these ONT reads. Exactly what you saw on the second video related to the fast QC quality testing of the mapping, which one showed you a pie graph followed by some depth and other things. Same thing can be done with ONT data also. Metagenomics protocols are also included within the program, and that allows you to use the sequence reads to generate a taxonomical profile, and these taxonomic profile can be charted, tabulated, and you can get the whole report of that. So that is also possible with the metagenomics platform, metagenomics protocols that's available within the platform. This is a program which I am running basically related to what is called the metagenomics. So where we create a bacterial repository, map the metagenomic reads, generate a taxonomy profile. I will just run it quickly because majority of the time I put it on this, I forgot to edit this, is on the reads basically. So once this is done, you can see that I have created the bacterial repository. Now I'm going to map the metagenomic reads of this bacterial repository using BWA-MEM mapper. You can see that that is what I showed you some time back. That is the ONT part of it, which is there, the Oxford Nanotech part of it, which I'm going to run this and get the mapping output. And this mapping output is read and put inside the repository itself, which is the bacterial repository. Once this is done, I create what is called the taxonomical profile of this. So the next step is basically creating the taxonomical profile. I will show you the output of this taxonomical profile as I run this. And once this is done, you get a taxonomic profile of this. And this is what is the taxonomic profile of the profile chart of the bacteria from the metagenomic read. So bacteria, what is the type of bacteria and how does it end to the level of the bacteria? Though this is the taxonomic profile. This is not a complex one, although little simplistic one, but of course we can help you run very complex one. So what we have seen till now, we can do the quality check in the reads. We can identify genes. We can identify SNPs. We can identify SNPs within genes. We can extract the insertions, deletions, and correlate the SNP with DB SNP or cleanware. You can compare genes and genomes. You saw in the variant part of it where you use cuff compare, cuff merge, and all of that. You can detect enrichment peaks. I did not show you this basically, but ChIP-seq will allow you to do that. You can do variant detection. A viral population analysis can be done, which I mentioned basically on the capability showcase. On the ONT part, I showed you a couple of components on the ONT, and also I showed you how we can do the mapping with the bacterial genome on the metagenomics part. I also showed you a component where you can use a CRISPR editing tool basically and then run the mapping programs. So this is what we have seen. So what do we infer from all this and what do we get from all of this? We need to understand the gene. Any polymorphism in the SNP within the gene will definitely reflect a different phenotypic association or a character with the external individual. What is that and how do we identify, quantify and profile that using the mass spectrometric techniques? So now you know the gene, you have the expression levels of the gene that you want to study, and you have the sample of that. You run through the mass spec. It can be MS1 or MS2. MS1 is called peptide mass fingerprinting. MS2 is called CID, collision induced dissociation, for confirmatory identification. So the whole set of programs are available here. Supports all types of instruments from Bruker to Thermo Fisher and Excalibur, and many of the other instruments is supported over here. The data from these instruments can be picked, and you can have your own reference database. By default, it uses an Uniprot Ref 100 database. But if you're looking at a subset of that, like say plant protein database or say a tissue related protein database, something like that, you can definitely extract that from Uniref and then include within this particular program so that it will be able to identify the precise protein that is obtained using the peptide mass fingerprinting data. Post that, you can do digestions and also identify the peptide. So you can identify the protein, you can do the confirmatory identification with MS2, you can do the quantification of the protein using SILAC or other methods. ICAT method, two methods are available. You can do differential profiling with the normal protein as well as the whatever is the sample or diseased protein respectively. So these things can be done with the proteomics. So what it gives as an output, it gives you, but please remember, you don't have to do all this. All of this are available within the program. When we ship you this program, you will get all of this. All you have to do is just add your parameter and add your input file and then run it. You can run it on the browser also, but this is a good way for people who want to innovate and see how it actually works and what we can offer as a remarkable difference in terms of the innovation that we offer. So you can do protein identification, and once the protein, and so you can see all the parameters are here. So we can use multiple database, single database, and you can use the cleavage or proteolytic digestion using trypsin, chymotrypsin, and any other proteolytic enzymes. Sorry. And if you have a custom cleavage site where you want to cleave at the tryptophan or some other site, you can also include that. And all this information is available, very self-explanatory with a detailed help which is available. So once you run this, you get a protein identification viewer, which will actually link you to the protein Uniprot ID, because we have uploaded on the database Uniref 100. So you have to do that way. Otherwise, if you want to include something else, you can do that also. So that accession number or the accession ID will be implicated on the output. So after the scan for this particular piece, what is the protein corresponding to that, along with the score and along with the matched peptide, will be given to you as an output. So if you want the peptide information for this, just click on one of the protein accession number and you will get the peptide information. So here we have done cleavage using trypsin basically, and this cleavage is what you get as an output of all the different peptides associated with that. If you click on this peptide of one of the peptide that matches to the protein, that peptide may be even a very contributing domain or bioactive domain of this particular protein if it is druggable basically. That also implies that. So there are many things that you can actually pop it out when you actually get a result out of this. So you have a peptide matching viewer, and when you click on that, that peptide in the sequence, what is the position of that will also be identified using this particular program. Or inside that protein, what are the different peptides? You can see that that is listed on below. This is the peptide matching viewer. And if you want to have evidence related to a particular peptide which is shown over there, you just click on that, and at the mass spec level, what is the peaks, the M by Z values associated with this peptide will be identified, and the list and the score will be provided to you on that. The fragmentation of an identified peptide will be showcased on the peptide matching viewer drilled down. So each of this fragmentation will be applied to a segment basically of the peptide, and that segment could be a region where there is a broken region or where there is a non-broken region. All of this information can be extracted after you do fragmentation matching. So this is how it looks like. Now, once you get the analyzed protein, you have to analyze the identified proteins. What you basically do is you want to analyze the protein after it is identified. So you have an alignment viewer where you can align and see whether there is a mutation or there is a change in the protein across each of the other protein sequences that is available for that species or that family of organisms or proteins. You can do that also. So this is at the proteomics level. Now we started with the reads, reference genome, de novo, ChIP-seq, RNA seek, all of that. Now we came at a place where we have identified the expression levels of these genes. Then we have identified what is the protein corresponding to that. And I said we can quantify the protein. Yeah, take it from me. We can quantify that protein expression level. Now the next step is to use chemistry basically to inhibit that protein or bring a molecule that acts as an agonist for this particular protein. So you need small molecules for that. The small molecules can be supported using what is called as the chemistry collection. So you have a set of readers, you set of writers, and different ways by which you can convert from one form to other. So you have one which can be from the SD format, you have sketch formats, you have RxN formats, you have Smileys format, you have PDB format, and so many. Then you have different manipulators and filters. You can do structure checking, you can do normalization, and you can do 2D and 3D coordinates. You can have your salt removals. All of these things can be done. Different calculators are available, which could be a physiochemical calculator, which could be a structural topological descriptors, and so on. And you can do searching and similarity analysis also. You can do different enumeration of these small molecules. So you can have a library of molecules, thematic libraries, focus libraries, and so on. You can have different database contents. You can access different web services for the synthetic chemistry part of it. You have a chemistry cartridge also associated with that. Then you have a molecular toolkit where you can use Perl, Java, or Pilot script to access and analyze them. Just give me a second. I'll just close the door and come. I think some sounds. Yeah, sorry, there was some sound. I just minimize that. Okay, so these are the different chemistry collections which are available on the this one on this particular program. And then I was also mentioning about imaging basically. So there are organizations which use different imaging programs. Say for example, when you are doing cell-based assays, or when you are using protein-based assays, there are certain images that comes out as an output basically. So there are several algorithms that is embedded within the program. So this is with respect to Pfizer actually was working on this, where they have integrated this program with Thermo Fisher store, that is Celomics, and a meniscus analysis can be done with this particular program. We have a company called Mendel Genetics. They actually use the different imaging techniques to improve the productivity and efficiency in agriculture with plate-based imaging application for seedlings basically. So this is how you actually identify them and then bookmark them and then identify the full saplings, that full leaves saplings that is available within the program. Okay, now we have done imaging, we have done gene expression analysis, we have done chemistry part of it. Now the most important part is protein modeling and simulation part of it. So this is where we use the protein target, we identify small molecules, we do protein-protein interaction, we do antibody modeling, formulation property, all of that. So before going into this, I just wanted to check if you or if anyone has any questions or anything that they want to ask before I proceed on.
มีท่านใดมีคำถามให้กับผู้บรรยายไหมคะระหว่างช่วงนี้สามารถที่จะถามได้นะคะสำหรับจีนNGS So far I don't see questions in the chat box.
Oh, questions are in the chat?
No, not yet.
Okay. Otherwise, we can even take up the question at the end of the session also. I'm okay with that.
ค่ะงั้นเดี๋ยวคำถามจะเป็นสุดท้ายก็ได้ Yeah, I think you can proceed, Anand.
Okay, fine. I'll do that. Yeah, thank you. So the next part is now, as I was mentioning, from the genes to the drugs was our topic. So from the systems biology to modeling and simulation. So we have completed the systems biology here. You saw that from the genes to the protein expression to the protein quantification and getting the protein. Okay, there is a part of also metabolite identification, which we showed you as a chemistry collection over here, which we can elaborate later also on that. And we have a metabolite collection also associated with the program, which uses a bioconductor program from R. Okay, so that is also there. Now it comes to the protein. Now you have the diseased protein or direct protein target, a pathogenic target of this particular protein. So whatever is the target, now you have to perform the modeling and simulation. So how do we do this? What are the different methods by which we can do this? Sorry. So on the drug discovery part, we have three sections. One is discovery of the target. Basically, it could be a protein target, the three targets that I mentioned. They could be the lead identification. It could be a natural compound, it could be a synthetic compound, it could be a microbiological compound, it could be an extract-based compound, anything. Then we have to do lead optimization. We have to optimize the lead compound. So these are the three techniques that is available within drug discovery at the preclinical or at the initial stage, which we'll be going through in this particular session. So this is a broad showcase of what is capable with Discovery Studio, and from the target identification to lead optimization. So what you see on the left hand side is related to protein target, homology modeling, X-ray, sequence analysis, antibody design, protein docking, protein engineering, and the center one is both small molecule as well as the macromolecule of the protein target. It could be simulation, protein developability, free energy simulation, scaffold hopping, virtual screening, fragment replacement, all of that. And the third one is the fourth one on the right hand side, which is applicable to only small molecules, basically ADME, toxicity, QSAR, library enumeration, and so on. So I will go through these details one by one. Why should you choose Dassault Systems BIOVIA? So it's not something that is in the market in the last 1 year, 2 year, 5 years, 6 year, 10 year, but it is in the market from 1978 onwards. So even 1980, even if we take it, it is around 35 years, 40 years around that. So it's 30 plus years of our presence in the market. So you can imagine the type of algorithm that we have provided to the research institutes, and many of them have achieved numerous things on that. So we have strongly integrated with some of the top universities and organizations. On the protein aggregation, we have integrated, we have collaborated with Massachusetts Institute of Technology and Novartis. Like that, we have integrated with Harvard, University of Massachusetts Medical School for protein docking, then NAMD Molecular Dynamics, basically with University of California, Illinois University, and then we have UCS of many of our programs. Modeler is a very big mark of that from Andrew Sally's lab. Like that, there are many integration that is done and present within the program. So when you want a program from any of these, you need to come to BIOVIA and then have this program installed on your system. First and foremost thing part of it is whatever we go and buy, the first thing we see is how does it look like? Does it look good? Nice shape, nice this one, how it is. So all that is done using enhanced graphics and display styles, what you see here. So these are the different display styles that you can achieve from your molecules that you're working on. Display is not just what you see in the eyes, but what information you can extract from the display is also important. So you can see that from the display that you saw earlier, you can extract the sequence information, the secondary structure confirmation information, the verification of the protein score, basically how good is the secondary structure. All that information can be verified. This is just some of them which I'm putting in, but a numerous set of information can be extracted from that. It's not only extraction of the information and visualization of images, but creating a surface or creating a boundary or creating an view to analyze further. That is called surface generation. If I want to see what is the electrostatic surface spread of the positive energy across my protein, what is the hydrophobic region of this my protein, I can't count each hydrophobic residue and tell this is the hydrophobic region. So I need a surface of that. I can't count each amino acid and tell that it is a positive protein or negative protein. So these are some of the drawbacks which were there. Maybe 20 years, 30 years back, we had no choice, but today we have such an advanced technology that we can just create a surface and tell what is the mapping of the positive region, the hydrophobic region, the acceptor region, the donor region, the positive ionizable region. All this information can be deciphered with a click of a button. So you can use simple surface generation and representation of them. So these are the different ways by which you visualize, extract information from the visualization, create your own idea within this, and then generate the information outside that using a surface generation approaches. There are different programs that's available within the program, within this particular suite, that allows you to perform a series of activity at the sequence analysis and the protein modeling stage. This is an extremely important and first step that you will be working on to understand the sequence and how do you model them. So we have homology modeling as one way by which we perform this. You can do mutant modeling. As I said sometime back from UCSF, we have modeler tool developed from Andrew Sally's lab, which is tightly integrated with this program. You can work on command line if you are a very sharp innovator of this and want to implement your command line ideas into that. That also can be done within the program. You can perform antibody modeling. You can do loop refinement with modeler or looper. There are two algorithms which is present. You can do side chain refinement using chi-rotor. There are 4 different rotamer programs that is available within the libraries that is available within the program. If you're working on membrane proteins, you can do transmembrane modeling. You can do protein design, basically mutation energy of binding and stability can be calculated. You can predict the disulfide bonds within the protein. You can do protein-protein docking, which involves even antibody-antigen docking also. Protein DNA docking can be done. Protein peptide docking can be done. All of this comes under the protein-protein docking, which comes from University of Massachusetts Medical School, Professor Zhang's laboratory. Spatial aggregation propensity is something which is gaining more and more importance today with the antibody vaccines and protein therapeutics, which is coming into the market. And their aggregation of the proteins is very important in ascertaining the stability of the protein when it is formulated. So that is extremely, extremely important, and we have a capability to work on that also. Once the sequence analysis is done, we can perform simulation and the X-ray analysis on that, because typically you try to use X-ray data as much as possible, other than using any other experimental data. If not, you try using homology model approach. So you can perform minimization with the simulation tool. So there are 4 different ways by which you can do minimization. One is smart minimizer, which is a combination of steepest descent and conjugate gradient. Then you have steepest descent, conjugate gradient, and adopted basis Newton-Raphson method, ABNR. Molecular dynamics can be done as a cascade where you have all steps together, or you can run individual steps. Explicit and implicit solvation can be achieved, and it can be an aqueous media, or it can be an implicit, which is based on Born-Oppenheimer approximation or Poisson-Boltzmann distribution, PBSA methods. Explicit also allows you to add ions and also add different cation and anion combinations. You can analyze the trajectory after the simulation is done. You can do preparation of the protein prior to doing all of this. Protein ionization and pKa predictions can also be done. You can treat the whole molecule molecular mechanical fashion and treat the active site of the protein in a quantum mechanical approach. The X-ray program does not allow you to solve the structure like CCP4 or other programs, SELEX for small molecule. But what it basically does is it allows you to generate electron density map. It allows you to pick water molecules and place ligands. It performs model refinement and validation. So these are the approaches by which X-rays can be used within the simulation program. Next comes docking. Docking is extremely important from the small molecule to the macromolecule interaction. So we have 3 different programs that you can use. One is called LibDock, and that is based on a virtual screening of millions of molecules. It takes around 1 to 2 seconds to run the docking of each molecule. Unlike CDocker, which is a molecular dynamics based docking program, the MD is based for the small molecule, not for the protein. But when you want to do flexible protein, it allows the flexibility of the protein along its side chain, basically, which adopts 3 different types of conformation, G+, G-, and trans conformation. So these are the different docking methods that you can use, and depending on the library size, you can use them. Typically use flexible docking for 1 or few compounds, and charm-based docking for up to 150 compounds, and a million plus compounds. Try using LibDock. So this is an optimized way of doing it. However, the program will not throw out an error if you're going to pump in large molecule, but it only takes longer time. You do docking when you have small molecules. You design when you don't have small molecule. You have the protein site, active site, but you don't know what are the molecules. But you want to design your own molecule. You do what is called the fragment-based design methods. So what you basically do is there is a fragment which is available within the program. So you go to PubChem or any other molecule which is database, download these compounds. Then you apply the fragmenter and it will generate fragments. So if you have 100 compounds, it will generate say 50 unique fragments. You can use these fragments and probe the binding site of these fragments using MCSS, which is called multiple copy simultaneous search, which is again coming from Martin Karplus lab, which is based on charm. So Martin Karplus was the only a computational chemist who got Nobel Prize for chemistry. So we are using that program, a Nobel laureates program within the solution. De novo receptor is based on interaction mapping. It maps and identifies and probes the binding site. Once you probe the binding site, if you get one fragment, you grow the fragment. If you get multiple fragments, join the fragment. If you get multiple fragments at two different locations, you link these fragments. So these are the approaches by which you can grow your whole structure. Once you grow the whole structure, you go to the patent database and find that the structure is already available. Keeping the interaction intact, you replace the core or do a scaffold hopping of this. So that is what we call it as replace fragment by modifying the scaffold. So the fragment based design program allows you to conform to all of these approaches with the chemistry person in mind. So the chemistry person will be able to suggest the type of reaction, and then you can embed within discovery this program. The next important one is pharmacophore. World has made significant achievements in this pharmacophore, where they have searched for databases, performed scaffold hopping, done virtual screening, done profiling of ligands. You have a small molecule, a natural compound, synthetic compound, you don't know what is the target for that. So you need to have a target database, and that is what the solution provides. And that gives you 4,38,000 different pharmacophore targets on which you can identify which is the perfect fit of your target. Okay, you can do it individually or you can do it in a group also. Then once you do that, you can do filtering of these poses, design fragments based on their activity, on their pharmacophores features. Then you can predict the activity based on the pharmacophores. The last point, predict activity based on pharmacophore, has gained more and more attention because we got a lot of compounds which came to the level of IC50, lower IC50 values with most of our customers, and many of them have published the results. Once you get a library of compounds, what you do, you cannot dump it or delete it. Basically, you need to retain that because it might be used for other target, because repurposing is something which is key. So you can do library design and analysis, and that could be a reaction based approach, or that could be an attachment point based or Marcus based approach. You can generate ligands which are not already present, unique of them. You can do different property calculations like ADME properties, toxicity predictions, xenotoxicity specifically. You can calculate the 2D and 3D properties of all of these compounds. You can do semi-empirical and DFT calculation of these molecules. Please bear in mind this, whatever I am telling you now is related to small molecules, not the proteins or peptides. You can identify a diverse set of molecules. You can cluster a set of molecules into a number of clusters. You can do similarity selection based on Tonimeter score, dye score, or cosine coefficients. You can do multiple objective Pareto optimization based on multiple properties. So these are the ways by which you develop a database and develop a library of their compounds. You identify a unique subset of them. You predict the property of them. You calculate the property of them. You find the diversity analysis of them, cluster them based on a property or on the chemical space. Then you can pick out those with certain specified property using Pareto optimization. So all the programs are available within this program. So now, after doing all this, you have identified 10 compounds. Wonderful, great. And these 10 compounds have to inhibit a protein. And this protein is a result of a gene polymorphism or a nucleotide polymorphism, single nucleotide polymorphism. And I have to inhibit this to overcome this particular disease. So what I do is what is called the structure activity relationship, QSR, to predict its activity, IC50 activity. And for categorical data, basically the classification data, I can use Bayesian and recursive partitioning. That's a Boolean, yes, no, it will tell. For continuous data, I can use recursive partitioning, genetic function approximation, partial least square, and multiple regression. So for continuous data is what I use to predict the IC50. We have many organizations, academics as well as industry, who have come to very close IC50 values when they run this enzyme assays, thereby saving money and time by not procuring all the compounds that comes as a result of a computational chemist's output, but rather a optimized set which they have purchased or synthesized by saving time and cost. So that's how QSAR works. The most important aspect as far as the regulatory is concerned is that you should have a set of compounds which is totally non-toxic. There should not be any toxicity associated with that. So we have a program called TopCat, that is toxicity based on computer-based technology. That K is called computer, KOMPUTER. That's how it was defined, computer-based technology. And this program assesses the genotoxicity as well as the irritation, skin irritation, then skin eye irritation, skin sensitization, moderate, severe, like that for each of these compounds, and follow specific guidelines. So these are available at the OECD guideline document, which is provided in the link over there. And this supports all the OECD guidelines in defining this machine learning model or a statistical model. And few other things that has been included in the program is explicit membrane-based molecular dynamic simulation. If you remember, I was telling you some time back that we can do membrane-based simulations, membrane transmembrane modeling. So if you're looking at a lipid membrane, a homolipid or heterolipid membrane, basically, if you're looking to build that, you can also do that. And this is what you will see in this lipid membranes which are available. So POPC, POP, DPPC, DMPC, and DLPC are the different types of lipids that are available, explicit bi-layer of lipids that is available that you can embed within the protein and then perform your molecular dynamics simulation to understand the activity. That is again a charm-based program, supports GPU as well as CPU, both whatever is your hardware, the infrastructure, based on that you can run it. The last part of it, not last part of it, there are a couple of more things, machine learning another one. But what I wanted to mention is the antibody modeling. I'll quickly go on this antibody modeling and show you what all we can do on the antibody modeling. You can do target selection on the antibody modeling. You can do target optimization and humanization. So there are VJ files and VC files available for humanization specifically. And those are available within the program that you can use. Different formulation properties can be predicted, like stability, at what pH, viscosity, ionization. All of this information can also be predicted. It can be based on pH dependent or pH independent, hydrophobe dependent, hydrophobic independent, and also identify different cleavage sites. Antibody humanization, you can search against light chain and heavy chain antibody sequences for humanizing mutations. And what you see on the regions is in the graph, some transgenic region from phage display regions, like that. Some polar hotspots will be available when you do humanization, and you can perform a mutation on them to study further. You can also work on bispecific antibodies, basically. I think bispecific is next slide. So rational antibody design, you can do bispecific antibody structure modeling. You can also perform de novo loop prediction and design your own components on that to predict the antibody structure. If you are looking at modeling bispecific monoclonal antibodies, where you have two specific target which form two part of the Y region, basically the Y large region, you can also do that, and there are a lot of examples associated with that. Removab is one of the approved bispecific monoclonal antibody which is existing, and I think it's from Eli Lilly, and that is what you have for the trifunctional monoclonal antibody, bispecific monoclonal antibody, which is there. So what you can do with this is you can do the expression studies, post-translational modification sites, sequence information, deamidation, glycosylation, all of that. Stability, aggregation spots, chemical stability, so on. Developability, solubility, viscosity, stability at pH, and so on. So all of these things can be done at the monoclonal level. The next part is protein aggregation and developability. As I said, program comes from MIT. Bernard Trout is the person's lab, and along with Novartis Laboratories, they performed this, came out with a unique algorithm, and that algorithm is part of Discovery Studio. So what takes you there is based on what is called the delta delta G, or the energy, basically. The lower the energy, the stable the compound. And if you're going to perform mutagenesis, how less energy is what you get is what you look at it with respect to the mutant as well as with the wild type.
So if you're looking at that, you can do virtual mutagenesis with that. Then you have in silico mutagenesis for protein binding affinity. So we have done certain studies with programs. This is a program called Rosetta, if I remember right. Yes. So the delta delta G program with Discovery Studio and other programs shows that the RMS error is 1.1, which is totally minimum after the mutation. So 380 mutations from 19 non-homologous protein were taken for experimentation, and all the study was done. And once you have designed your monoclonal antibody or a protein therapeutic, you need to know what are the different formulation property associated with that. And this is the program that allows you to identify the formulation properties like developability, like aggregation, solubility, pH stability, and so on. Then we have the predicted formulation properties as below. So you can see that we have predicted formulation properties which are available. So you can see net charge, isoelectric point, pH of maximum stability, and so on. So like this, all of these predict formulation properties are available, and this information is available when you run predict formulation property as this protocol. So just to summarize on this, from protein sequence, you can identify post-translational modification sites, calculate biophysical properties, annotate antibody domains, then you can align and compare sequences. From the protein structures, you can generate structures, use a pH-dependent protein property calculation, predict developability, aggregation propensity, surface patches, optimize the binding affinity, pH-based, non-pH-based dipole moment with dipole moment. The next part is a short one on data science and drug discovery, because today we see that Essentia is one organization which is working. Essentia is not one organization, but one successful organization which got some results of a drug that was developed using AI approach and is in the clinical trials in Japan. So on similar lines, if you're going to use data science for drug discovery, how it helps. So this is what we have, a solution, and this solution is called as generative therapeutics design. You can have a look on that and see how it takes up to 5 years for 4,000 compounds. Then, but these 4,000 compounds across different rounds have different issues, and these issues can be addressed using what is called a machine learning approach, and that is based on target product profile, TPP. That is what we call it as, and that is correlated to the activity, and you can see the activity levels of each of these compounds. So like this, you can have an automated fashion by which you can generate and predict the IC50 values. You can also do a manual human intervention to understand the target and the profiles of these compounds. Although it's machine learning, you can do all of these activities, predict the toxicity profile, ADME profile of these compounds that you shortlisted, and then take to the next part of the drug discovery process. If you are working specifically on where you want to use a machine learning approach to classify certain specific image objects and then annotate them, and then you want to say that you want to search it across all the other ones and say what is the unique part of it, you can do that using an image annotator. The program will be shipped along with this image annotator, where you can do your own machine learning on the images. This is based on a Gaussian classification or k-means classification that you can achieve it. So for general protocols are available. If you're going to use certain unique images, we can help you and train you to do this, and you can start doing it on your own. We also have another opportunity for imaging to identify melanoma, basically a benign or malignant tumor in a set of sample images, basically. So what you see here is when you run this program, it generates a confusion matrix to give you the true positives and the true negatives. Additionally, it also gives you the whole information related to the skin lesions, which is designated as benign or malignant. The level of accuracy in all of these programs is 98% plus, because it uses a very strong algorithm to classify based on these images. This is a recent publication with machine learning for antibody, which was done from Bernard Trout, that is MIT. And this is the title of that, you can have a look on that, and they have used exclusively our solution to come out and propose formulation excipients to move forward with the development. This is another publication from a pharma company where they have identified the sites of antibody degradation and find the main characteristics that distinguish the stable asparagine and aspartic acid residues in the variable region of monoclonal antibodies. So this is what is another publication from the pharma. Now I want to add another one part to the whole lot, because yes, you would be doing a lot of experiments, but all these experiments you would either record it on paper or record it on your notebook. If you have a synthetic chemist or a microbiologist or biotechnologist, whatever it is. But when you do a publication and when you want to dig out an information or address a query that comes from the attorneys or from the reviewer, basically you need to search for this by using different search techniques, which will eat all your time as well as the level of accuracy which you will let. So we can offer you a flexible way by which you can record all your experiments in a nice notebook sort of thing. Very simple to notebook. It's very similar to notebook, and that is totally compliant as per the guidelines. And tomorrow, if you want to extract the information, all of them is available. Why? Because three Swedish universities, University of Ghent, University of Strasbourg, and Karolinska University, have 10,000 users who use this notebook and who collaborate across this platform for experiments. So a person from chemistry lab can collaborate with computational chemistry, with microbiology, and so on, and every one of them works collaboratively, come out with innovation. So this is how they work on, and this definitely can be thought about in your university or academic institute. So what do you get if you use this? You get a high productivity. You save time every day, not writing the experiment, not cloning the experiment, not pasting results on that. All of this is gone, and the quality of the records is very good, can be retrieved at any point of time, can be showcased at any point of time. Your principal investigator will be able to approve it on the system itself, so you don't have to carry the notebook and take the signature from him. All of this can be done using the BIOVIA notebook to digitalize research labs. Last but not the least, I also want to introduce you to what is called the chemistry sketcher. For chemistry and for biologists and microbiologists, we have a chemistry sketcher that is available. You can go to the website, you can register on the website if you are an academic institution, and then you can see what is chemistry structure and just try using a chemistry structure where you can use protein sequences, peptides, and chemical structures and perform modifications and represent in a variety of formats also within the chemistry structure tools. So what I want to conclude is pharmacogenomics in pharmaceutical chemistry is potential tool, and we have to maximize the benefit on that. And currently these pharmacogenetic methods are used worldwide for assessing the safety profile of the drugs, personalized medicine, pharmacogenomics, and so on. So as the need arises, identification of new biomarkers, and then patenting of these biomarkers specifically, and then taking the ownership of these biomarkers, which might be a druggable target, is a key important aspects for all of this. And in COVID-19, you would have seen the spike protein as one of the biomarker that came up. Similar to that, many diseases, many biomarkers can come up. So these can be utilized by academic and pharmaceutical industry for drug development processes. So we need to gradually but surely include pharmacogenomic study in drug discovery and development. That will not only cause substantial reduction in expenses, but also ensure a safe clinical trial and reduce the number of failures. Because 20 years back, 30 years back, the number of drugs that came into the market was phenomenally higher compared to what is coming into the market today. Possibly because a lot of compounds enter at the initial stage, but when it goes to the stage clinical trials stage 4 or stage 3, it substantially fails. So if we could identify what is the reason for that using these type of approaches, we can definitely reduce these failures. That is what I want to conclude.